We'll see if it works. That. Yeah, so the focus of my talk is, as Elina indicated, storage of memories, because we all know that we forget things. There are little things and larger things that we shouldn't forget, but we just forget them anyway. And then there are things that we cannot forget. Often those are emotional events or episodes, like the recent inauguration of Donald Trump. So how come we can't forget things, and how come things are stored for such a long time? That's something that we know very little about. We know a lot about how memories are formed, how they are encoded, and also kind of the short-term memory storage. But we know very little about the mechanisms to support these long-term memories. And during learning and also brain plasticity, what happens is that there is some degree of rewiring. So we say that if neurons fire together, that means that if they send a neural signal together, then they will wire together, so they will strengthen their connectivity. And we also know that in young individuals, both in animals and also in humans, there is a much higher degree of plasticity in the young, in the early ages than it is in adults. We are still able to learn, but the degree of plasticity is much less. And then we ask if these mechanisms that limit the brain plasticity in the adults, could they also be kind of a framework to support long-term memories? Because, as I said, these changes that occurs, they depend on the proteins. But the proteins, the turnover rates of those proteins, they are much shorter than the how long these memories lasts. So that's kind of it's a little bit unexplored territory. At least it's unexplored, or we don't understand it. But this is an image of just a histology image. That means kind of you see these red little dots. Those are neurons in a rat brain. And what we see is that during development, then suddenly in this transition zone between the young brain and the more adult brain, suddenly what appears is these very dense extracellular matrix molecules that enwraps some of these neurons. And if you have a closer look on these structures, you'll see that it has kind of a mesh structure, so there are little holes in the structure. And these holes are where these synapses, these contact points, are between neurons. So what we ask is if these structures actually, which appear at a time when the brain plasticity is being lowered, if those could also be kind of a physical framework for long-term memory storage. And in order to test that, we need kind of an ideal model system. And what we use is fear memories. Because fear memories, they are very easily induced, and they are very robust, so they are long-lasting. Also, interestingly, is that they are dependent on these very deep, evolutionary old brain structures. That means that there may be some transitional relevance or translational relevance from animals to humans studying these kinds of behaviors. Also, convenient for us is that somehow they are transported or transferred into the cortex. That makes them very accessible for our manipulations. So what Mattis and Elise and Christian did in our lab was that they trained animals in such a paradigm. So they placed a rat in a box and then presented a neutral stimulus that would be a light. It's not scary for the animal, but they associated that to a kind of a weak electrical foot shock. It would be so that it would be unpleasant for you to touch, but you wouldn't be scared. But these animals, they get extremely scared, and they report that back to us by sitting completely still. So if we test them 1 month later and only present the light, they will be kind of this freezing position, standing still. So this is also quantified here. It's like each dot would be one animal. So before training, they are not afraid. This is kind of indication of how afraid they were. And then after 1 month, 50% of the time, they sit there completely still. So they know, they remember this association. So then what the students did, they then trained the animal for 1 day and then waited for 20 days so that the memory was remote. And then they removed this structure. And they did it only in a tiny, tiny part of the cortex where we think that they should be stored. And then they tested it then after 30 days. So what they found is first just a recap of that the control animals, they do remember. But if they remove this perineuronal nets, as we call them, it's kind of an erasure of the memory. They can't recall anymore. And through a lot of different control experiments, we are pretty certain now that these nets are responsible for stabilizing the neural network that is involved in this recall of the fear memories. But the kind of the story that I presented so far, it's a descriptive story, so we can't really claim that we understand the underlying mechanisms. And these are huge questions. And the cool thing in neuroscience, and also all fields of neuroscience, is that there is a huge technology development, meaning that we could record almost anything, and in any way we could manipulate it. So this is from a zebrafish. It's kind of all the neurons in a zebrafish imaged at the same time. We could place hundreds of electrodes into the rat brains and during these behaviors and get immensely amount of data. But the problem is that we can't kind of make use of those data just by looking at it. And also, even though we could kind of record all of this really cool thing, it would always be only a glimpse of what actually happens in this process, which kind of involves all these levels, from the genes to the proteins, all up to behavior. And there will never be a device that could measure all of these processes at the same time. And that's why we have joined forces with these physicists and mathematicians for them to make more computational models, which could then explain actually what we record, the underlying kind of physics behind what we record, and they can fill in the gaps that we can't record, and also they could link these different scales. So that's what we have tried to do in this research initiative that Elina mentioned, Simpla. We joined forces between this experimental work that I represent and then computational physics and computer science. But this really is kind of jumping out of your comfort zone, because you will have to talk to people that don't understand you don't understand them. So there will be kind of a lot of time spent just on trying to understand each other, which is rewarding, but you will be you have to be willing to invest in this. But what then they then do is that they could make very advanced mathematical models of these neurons, and then they could start to ask, well, how do they communicate? How could the different cell populations contribute to this, and how do they communicate, like Espen and Mikkel has done here? And then they could go further, so they could say, well, we connect all these in a kind of anatomically correct way, as we understand the brain, and then they could place a virtual electrode, while we usually implant them in a rat brain, and then they could see, well, how does these kind of mathematically model neurons respond? And then they could look at this population response of all the neurons, and that's actually what you will measure with the EEG signal. So EEG recordings, that is that you place a lot of electrodes on top of the human scalp, and by that you record some neural activity. And this has been done for years and years and years. But the thing is that we really don't understand the relationship between kind of the individual neurons. How do they actually contribute to the signal that we record? And that's what Solveig Næss in our group is focusing on. So she's using those mathematical models of a neuron, and then she uses different fields of physics to understand how they would respond, and then she could kind of connect those and place those into a model to understand, well, how would that signal relate to the EEG recordings that she actually or that we record in the clinical situation. So I think that this is kind of ongoing work. We have this descriptive, very exciting finding from the experiments, but now we try to combine with the experiment or the computational models to really understand the underlying principles of how these processes come about. And then I would just give a little teaser to these 4 computational physics students in our group who has developed an educational app so that you yourself can play around and connect all of these neurons and play around with the parameters. So this was just the app that they have done made have already like almost 30,000 downloads. So it seems to be meeting not a need, but at least an interest from the community. So I would just like to end by thanking all of these fantastic candidates. That's kind of altogether is this center of integrated neuroplasticity. and perhaps especially to the people that contribute to the work today, Elise, Christian, and Mattis, and Mikkel, and Espen, and Solveig. So, thank you.
Thank you very much, Marianne. That was a very interesting lecture. I have some very short questions for you. Now that we talk a lot about the fake news, but are there fake memories, and how are these stored?
So there are fake memories, kind of, because so I've not studied those myself, but one knows from, say, if you are told that this and this happens, and then you could relate other things to that, and then suddenly you start to believe them. And likely they are stored in the same way as our kind of real memories. Likely, but it's kind of not that well understood, but likely the biological mechanisms for storing memories would be the same.
So I could sort of create my own happy childhood.
Yeah, that you do, because people report that they could kind of come up with memories from before they are 2 years old. That's extremely unlikely, because the brain is not developed in such a way that you could store the memories at that age. But from pictures from people that tell you, then you create these very live images of how it happened, and that's kind of the way one thinks that you could remember from your early childhood, which you actually don't perhaps remember.
Cool. But then when you said that fear induces very strong memories that you remember for a long time, I mean, when I was a student, would that be sort of a way of teaching people for a way that you really would remember for an exam, if you like induce fear into teaching?
I think that the problem is that then kind of everything is so focused on that kind of threatening experience, so you don't remember anything else. And it's also shown that kind of a positive reward way of learning is more efficient to learn things than a negative or a punishment way of
well, that's a bit relieving to hear.
Yeah, I think that at least we should bring that into our own teaching.
Good. We have a small present for you as a thank you for telling us about a little bit about the brain and how the brain works. So this is coffee cup that will store your coffee, not the memories of it, but the heat, and reserve the heat for a longer time. So thank you very much.
So thank you very much. So I'm a music researcher, and you may wonder what a music researcher is doing at a life science event. And I'll try to explain that during this talk. So my research is about trying to understand musical experiences, like this one. What happens in a performance context like this? What happens with the performer? What happens with the audience? And this is a classical setting where you sit down, but we also study different types of dancing to music, like in this context. What happens here with the performer and also with the people dancing? And what does all of this have to do with this? An infant lying on a mattress. Well, in our research, we have gone from developing tools for basic humanities research, and we are now in the process of developing what is called a computer-based infant movement assessment, and that is now in clinical testing in hospitals around the world. But to explain you how we got to this, I need to kind of wind back a little bit and go back to 2005. And at the time, I was doing my PhD, and I was sitting in a lab in a cold winter in Montreal at McGill University. I was really trying to figure out how can I analyze a bunch of these type of video recordings. So we had done a series of recordings of people dancing and moving spontaneously to musical sound. And we were interested in trying to figure out what's going on. What's happening? How can we make some kind of representation? How can we extract something meaningful from this? Okay? So we're talking about human bodily movement in time and space. And the challenge here is really to figure out how can we represent this in a way that so that we can compare the movement to the musical sound and other types of musical features. So I started developing a set of tools and techniques for doing this, and started out with something called motion history images, which is basically just a kind of a feedback representation of the video that you can see. So you start with the original video image on top there, and then you have different types of variations on this, where you kind of get a representation of the motion over time. So you can consider trajectories in a way similar to how we also experience and see a motion sequence. But again, these are fairly short periods of time that you can visualize with this type of method. You can, of course, also make a static image of this, where you can kind of see the traces again of how a dancer was moving in time and space. Or you can create something we call a motion average image, where you kind of look at the global activity over time. This is from a 5-minute dance sequence. And this is not very useful, although it kind of gives you at least some sense that the dancer was mainly standing still on the floor and kind of moving the arms around. But as a music researcher, I'm interested in time and how movement and sound develop and change over time. So the temporal dimension is crucial to what we're doing. So for that reason, I ended up developing something called motion grams. And this is both a kind of a general method, but we also developed software tools for this. And motion grams, they look like this. We start with a motion image, which is just a reduction of the frame differencing of a video stream over time. Then we collapse it and we can plot it on either side. So it's basically just a reduction of the original video. We don't need any markers or anything. It's just a plain video recording, and you can get a representation like this over time. So if you look at a sequence a motion gram based on the video recording I showed you earlier, this is the entire 5-minute dance sequence you started seeing the beginning of earlier. So in this display here, you start here, and you go over here. And you can get an idea of the spatial temporal development over time by looking at this. We can zoom in a little bit so we can see more details from the first 40 seconds of this. And again, you can start to see that this may be useful for understanding more of how the body changes over time temporally. Skipping ahead a little bit, I was invited to present this research at this annual event that the Norwegian Research Council hosts at the Oslo Concert Hall. And I came there with a dancer, and we was actually doing this. We were even playing this and using this live, because you can do this while a person is dancing. And it's also possible to create music again from this, so it's kind of going both in and out of analysis and synthesis. The interesting thing about presenting to a big audience like I had there was that there's always someone that will come up to you afterwards and say, "Oh, that was a good idea. Can I try that on my stuff?" And that also happened in this case. So moving forwards a little bit, I ended up in Trondheim at St. Olavs University Hospital, working with Lars Adde, who's a physiotherapist there. And his main research topic was that of understanding how children move. And he's particularly interested in detecting cerebral palsy in preterm infants, because if you can detect cerebral palsy, or at least the infants that are in the risk zone of developing cerebral palsy at a very early stage, you can also start physical intervention very early on. That again may help prevent further development of the disease. So a few years later now, we have been collaborating for 7 years now, more or less. And there's now this product that we are developing called Seaman, which is a suitcase, as you see here, standardized setup, where you have a mattress, and there's a camera standing here. You can't really see the camera. And then the infant will be lying on this mattress and will be filmed with a video camera. There's nothing you need to put on the body at all, just being filmed for a couple of minutes. And then we run the software analysis on this, and we can then, with a fairly high certainty, be able to say whether this child is in the risk zone of developing cerebral palsy or not. And then it can be followed up further on. The positive thing about this is that today, there are only specialists. There are only a few special physiotherapists that can do this type of detection today. So most infants, and particularly the preterm ones, are not screened at all today. So with this tool, we can basically have this at every hospital around the world, and also be able to follow up the children that really need to be followed up. And the key for this to happen is really that we started with the software that I developed for studying the dance movements, where we look at the rhythmicity of the movements and the temporal development of this. And we found that when we were looking at the infants, this is a normal infant here, and this is an abnormal behavior where it was later discovered cerebral palsy. And you can't really see it very clearly from the graph here, but based on these graphs and also from some more quantitative analysis that we're doing on top of this, we are able to say with fairly high certainty whether these infants will develop cerebral palsy or not. Now, the interesting thing here for me as a music researcher, because I'm staying on as a music researcher, is that something that started out really as basic research in the humanities have ended up as innovation in medicine. And that's kind of just proving that supporting and funding basic research is important. And then, of course, we are very happy now to have received funding for Ritmo, our new Center of Excellence, where we are teaming up with musicologists, psychologists, and informatics researchers. And we're going to study not the meaning of life, but the meaning of rhythm in human life, and really try to uncover more of both the cultural, the historical, aesthetical, perceptual, cognitive, and technological aspects of rhythm, nothing less. So I think I conclude there. Thank you very much for your attention.
Thank you very much for a very interesting lecture. I actually, I can reveal this now. I used to dance. I used to be a dancer. I found this very, very interesting, especially to look at how we move to music. But what struck me is that we move very differently in very different situations. And I know for myself that my movements are different from, when I'm happy, or if I'm sad, or maybe if I'm in love, I would be moving differently. Is there any limit to what we could measure with this kind of technology? I mean, what would you be able to detect, not only when it comes to disease, but...
Well, that's a good question. We have been looking at fairly large-scale body movement, more kind of actions or kind of things that you do on purpose. But I also have a new research project now where I'm looking at people standing still, and where the whole point is really to try to reveal something about the unconscious level based on looking at just the standstill or the micromotion of people. We do see that there's a lot of things we can actually see in this micromotion that you are not aware of yourself. So be aware. So
me standing here is actually me revealing myself, quite trying to stand still, not...
Absolutely. I mean, there's a lot of... one thing is the micro-expressions in your face, but also just the micro-expressions that we measure, for example, just by the head movement you have, can say something about your state of mind. Have you find anything, like if I do like this, or what could you extract from my micro-movements at this point? Well, we are still working on a lot of analysis on this, so we don't have any kind of concrete findings yet of how these correlations are, but that will come in a couple of years.
What so far in your studies, what surprised you the most to find from studying movement and music?
Well, I guess the most surprising finding is that people are actually more similar than they think, and I guess that might be an obvious finding. But if you ask people to move to musical sounds, they often do more or less the same thing. So if you hear a pitch going up, you move upwards. When you hear a musical beat, you will start nodding your head. I mean, we're doing a lot of these studies in our lab, and people always come in, they say they are so different, they listen to all sorts of music, etc. But it's, in fact, they are doing more or less the same thing, even when they are alone in the lab.
Nice. Nice to know. Thank you very much for your lecture. And as a small token of our gratitude, this is a cup coffee cup to keep your coffee warm when you're dancing in your lab, or looking at other people dancing in your lab. So thank you very much.
Thank you.
Before we finish, I want to thank you all for coming here today. And remind you that the next Life Science Lunch will be the 26th of April. So at 12 o'clock, 12:15, same time, same place, 26th of April. Thank you very much for your attention.